Today we are learn interesting topic rDNA technology, recombinant DNA technology. Okay, recombinant DNA technology or rDNA technology. Okay, genetic engineering primarily involves that manipulation of genetic material. Okay, genetic material means DNA to achieve the desired goal in a predetermined way. Okay. Some other terms are also in common use to describe genetic engineering. Okay. That is called recombinant DNA technology. Okay. What do you learn from this course? Introduction to rDNA. Okay. Next, definition, steps, applications, and limitations. Okay. Introduction. Recombinant DNA technology is the joining together of DNA molecules from two different species. Okay, the recombinant DNA molecule is inserted into a host organism to produce new genetic combinations. Okay, recombinant DNA technology is the joining together of DNA molecules from two different species. Two different species means example human beings, mammals, okay, your interest genes, okay. The recombinant DNA molecule is inserted into a host organism means host organisms means after recombinant DNA molecule will be ready. Okay. Next, we transform into the host organism that is called E. coli, Escherichia coliform bacteria. Okay. To produce new genetic combinations that is called recombinant DNA molecule. Okay. Why we need this and what is the purpose in daily life scenarios? It's add value to science, medicine, agriculture, and industry solutions. Okay. The idea of recombinant DNA was first proposed by Peter Lowen, okay, a graduate student of Professor Dale Kaiser in the biochemistry department at Stanford University Medical School. Okay, Stanford University applied for a US patent on recombinant DNA in 1974. Okay, inventors as Herbert W. Boyer. Okay, he's a professor at the University of California, San Francisco, and Stanley and Cohen. Okay, this patent was awarded in 1980. Okay, the patent was awarded in 1980. Okay, history of recombinant DNA technology. The present today DNA technology has its roots in the experiments performed by Boyer and Cohen in 1970. 73. Okay. In their experiments, they successfully recombinate two plasmids, that is PSC 101 and PSC 102. Okay. And cloned the new plasmid in E. coli. Okay. Plasmid PSC 101 possess a gene resistant to antibiotic tetracycline, while plasmid PSC 102 contains a gene resistant to another antibiotic kanamycin. The newly developed recombinant plasmid, when incorporated into the bacteria, exhibited resistance to both the antibiotics tetracycline and kanamycin. Okay. Next, the second set of experiment of Boyer and Cohen were more orga- organized. Okay, a gene encoding a protein uh, requires to from RNA. RNA means ribosomal RNA. Okay, rRNA was isolated from the cell of African clawed frog. Okay, frog scientific name Xenophis levis. Okay, this is the frog scientific name. Okay, by use of restriction endonuclease enzyme. Okay, that is called EcoR1. The same enzyme was used to cut open plasmid PSC101 DNA. Okay, frog DNA fragments and plasmid DNA fragments were mixed, and pairing occurred between the complementary base pairs. Okay, by the addition of the enzyme DNA ligase, a recombinant plasmid DNA was developed. Okay, the new plasmid, when introduced into E. coli and grown on a nutrient medium, resulted in the production of an extra protein that is called the frog protein. Okay, thus the genes of a frog could be successfully transplanted. This is history of recombinant DNA technology. Okay, the genes of frog could be successfully transplanted and expressed in E. coli. This made the real beginning of modern DNA technology and foundation for the present day molecular biotechnology. Okay, some biotechnologists who admire Boyer Cohen experiments divide the subject into two chronological categories. Okay, BBC and ABC. Okay, BBC means biotechnology before Boyer and Cohen. Okay, uh, next ABC means biotechnology after Boyer and Cohen. Okay, more information on the historical developments of genetic engineering and biotechnology of Rdna technology. Okay, next 1 minute. This is a brief history of Rdna technology. Okay, first experiment by frog. Next, Rdna technology is developed. First, definition. Okay, recombinant DNA technology definition. The joining together of DNA molecules from different organisms and inserting it into a host organism to produce new genetic combination that are of value to science, medicine, agriculture, and industry. Okay, a series of procedures that are used to join together Okay, DNA segments. A recombinant DNA molecule is constructed from segments of two or more different DNA molecules. Okay, it is the technology to produce an artificial DNA molecule by combining two or more fragments of DNA which are not necessarily associated with each other. Okay, this is definition of recombinant DNA. Okay, RDNA, recombinant DNA definition. Next, clone. Okay, gene cloning. First clone. Clone is a molecule, okay, cell or organism that was produced from another single entity. Okay, clone means colony. Colony means bacterial colony. Okay, that is the recombinant clone or colony. Okay, I explain brief next sessions. Okay, made possible by the discovery of restriction enzymes. Okay, plasmid DNA vectors. What is the restriction enzymes? DNA cutting enzymes. Okay, molecular scissors. Restriction enzymes, it is the cut the DNA fragments. It is like a scissors. Okay, restriction enzymes isolated from the bacterial species. Okay, some are commercially also available. Okay, next, plasmid DNA vector. Plasmid means it is the circular, double-stranded, extra chromosomal, self-replicating DNA molecule that is called plasmid DNA vector. Okay, can be manipulated to carry and clone other pieces of DNA. Okay, look at here. Restriction enzymes already I told. Okay, primarily found in bacteria. They use these for defense. Cut DNA by cleaving the phosphodiester bond. Okay, that joins adjacent nucleotides in a DNA strand. Bind to recognize and cut DNA within specific sequences of bases called restriction site. Okay, each restriction site is a palindrome. Read same forward and backward on opposite strands of DNA. Palindrome means six nucleotide base pair sequence. Six nucleotide base pair sequences means adenine, adenine, guanine, cytosine, thymine. Okay, repeat: adenine, guanine, cytosine, thymine. Any nucleotide base pair sequences, it is called nucleotide base pair sequences. Okay, only six digits. Okay, for example, only this is example: guanine, cytosine, thymine. Okay, six nucleotide base pair sequence, that is palindrome sequence. Okay, there are four or six base pairs cut because they recognize restriction sites with a sequence of four or six nucleotides. Okay, that is called palindrome sequence. Okay, are you understand? Okay. Once again, I repeat. This is only introduction of RDNA technology. RDNA technology. Okay. Genetic engineering primarily involves the manipulation of genetic material. Okay, genetic metabolism means DNA, deoxyribonucleic acid. Okay, DNA. To achieve the desired goal in a predetermined way. Some of the terms are also in common use to describe genetic engineering. Okay, genetic engineering, gene manipulations, recombinant DNA technology, gene cloning, or molecular cloning, genetic modifications, new genetics. Okay, these are in a recombinant DNA technology. Okay, introduction. Recombinant DNA technology is the joining together of DNA molecules from two different species. Okay, two different species means bacterial species or human DNA fragments. Okay, mammals. Okay, any your interested genes. Both are two different species of DNA fragments. That is called two different species of DNA fragments. Okay, the recombinant DNA molecule is inserted into a host organism to produce new genetic combinations. Okay, the recombinant DNA molecule is inserted into the host organism. Host organism means E. coli, Escherichia coliform bacteria, to produce new genetic combination. Okay, that is called clone. Okay, why we need this and what is the purpose in daily life scenarios? Science, medicine, agriculture, and industry solutions. It is a very important role in RDNA technology. Okay, the idea of recombinant DNA was first proposed by Peter Lowell, a graduate student of Professor Dalcaster in the biochemistry department at Stanford University. Okay, Stanford University applied for a US patent on recombinant DNA in 1974. Okay, listing inventors as Herbert W. Boyer. Okay, he's very important scientist, professor of professor at the University of California, San Francisco, and Stanley and Cohen, professor at Stanford University. This patent was awarded in 1980. Okay, Boyer and Cohen. They successfully recombinant plasmids. Okay, ABC. Okay, ABC means biotechnology before Boyer and Cohen. BBC means biotechnology before Boyer and Cohen. ABC means biotechnology after Boyer and Cohen. Okay, Boyer and Cohen in 1970 in their experiments. The present DNA technology has its roots in the experiments performed by the Boyer and Cohen in 1973 in their experiments. Okay, they successfully recombined two plasmids, okay, PSC 101 and PSC 102. Definition: the joining together of DNA molecules from different organisms and inserting it into a host organisms to produce new genetic combinations that are of value to science, medicine, agriculture, and industry. Okay, recombinant the a series of a procedure that are used to join together recombined DNA segments. A recombinant DNA molecule is constructed from segments of two or more different DNA molecules. It is the technology to produce an artificial DNA molecule by combining two or more fragments of DNA which are not necessarily associated with each other. Okay. Engineer is a person who designs, constructs, example, bridges, canals, railways, okay, and manipulates according to a set plan. Okay, the term genetic engineering may be appropriate for an individual who is involved in genetic manipulations. Okay, the genetic engineers toolkit or molecular tools, namely the enzymes most commonly used in recombinant DNA experiments. Okay, that is called restriction enzymes, ligase enzymes. Okay, clone. Look at here. Clone a molecule. Okay, a cell or organism that was produced from another single entity. Okay, clone means colony, like a colony. Okay, colony means bacterial colony, bacterial species colony. Okay, next restriction enzymes. It is very very important enzymes. Next ligase enzymes also very important. Okay, restriction enzymes, DNA cutting enzymes, molecular scissors. Okay, these restriction enzymes isolated from the bacterial species. Okay, example eco R1. Okay, SMA1. This is called bacterial species. Okay, okay. These restriction enzymes with sources, recognition, sequences, and the products is formed. Okay, example eco R1, Escherichia coliform bacteria. Okay, isolated from the eco R1. This is the enzyme. Okay, these enzyme isolated from the one bacterial species that is called Escherichia coliform bacteria. Okay, next bomb H1. This is the restriction enzyme. This is isolated from the Bacillus amelocaceus. Okay, bomb H1 restriction enzyme isolated from the Bacillus amelocaceus. Okay, these are restriction enzymes. Okay, next one HAE3. Okay, Haemophilus aegyptus. Okay, this is isolated from the Haemophilus aegyptus bacterial species HAE3 restriction enzyme. Next, HIND3. HIND3 is the restriction enzyme. This is isolated from the Haemophilus influenza. This is one type of bacterial species. Next, not one. Nocardiae otidis. Okay, this is bacterial species isolated from the not one restriction enzyme. These are isolated from the bacterial species. Some are commercially also available. Once again, I repeat restriction enzymes examples. Okay, EcoR1, BAM H1, HAE3, HIND3, not one. Okay, these are restriction enzymes. Okay, these restriction enzymes isolated from the bacterial species. Different restriction enzymes is there. These enzymes isolated from the different species of different bacterial species. Okay, are you understand? Next, ligase enzyme is there. Okay, 1 minute. Restriction endonucleases are one of the most important groups of enzymes for the manipulation of DNA. These are the bacterial enzymes that can cut or split DNA from any source, okay, any source of DNA at specific sites. They were first discovered in E. coli, restricting the replication of bacteriophages. Okay, by cutting the viral DNA, the host E. coli DNA is protected from cleavage by addition of methyl groups. Okay, thus the enzymes that restrict the viral replication are known as restriction enzymes or restriction endonucleases. Okay, are you understand? Hundreds of restriction endonucleases have been isolated from bacterial species, and some of them are commercially also available. Okay, the progress and growth of biotechnology is unimaginable without the availability of restriction enzymes. Okay, next, plasmid DNA vectors. Okay, plasmids, it is the circular, double-stranded, extra chromosomal, self-replicating DNA molecule. It is called plasmid. Okay, it is a vector, like just like a vehicle. Okay, these plasmid DNA vectors, okay, vectors are the DNA molecules which can carry a foreign DNA fragment to be cloned. They are self-replicating in an appropriate host cell. Okay. The most important vectors are plasmids, bacteriophages, cosmids, and plasmids. Okay, PBR 322, PUC 19. Number of plasmid DNA vectors is there. Okay, it is just like a vehicle. Genes transfer from one species to another species. Okay, okay. Next, cut DNA by cleaving the phosphodiester bond that joins adjacent nucleotide in DNA strand. Okay, cut DNA with specific sequence of bases called restriction site. Restriction site is a palindrome. Palindrome means six nucleotide base pair sequence. Adenine binding with the thymine. Okay. Guanine binding with the cytosine. Already you know. Okay. This is the nucleotide base pair sequences. Reads same forward and backward on opposite strands of DNA. Okay. These particular six nucleotide base pair sequence called palindrome sequence. That is the restriction site. There are four or six base pairs cut as because they recognize restriction site with a sequence of four or six nucleotide base pair sequences. Okay, next, ligase enzymes. Okay, what is the ligase enzymes? ligase enzymes. It is also very important role in our DNA technology. Okay, this is not mentioned, but it is very important. DNA ligases. Okay, the cut DNA fragments are covalently joining together by DNA ligase. Okay, ACE means enzymes, restriction enzymes. Okay, restriction enzymes. Next, ligase also enzymes. Okay, these enzymes were originally isolated from the viruses. Okay, they also occur in E. coli and eukaryotic cells. DNA ligases actively participated in cellular DNA repair process. Okay, the action of DNA ligases is absolutely required to permanently hold DNA pieces. This is so since the hydrogen bonds formed between the complementary bases. Okay, complementary bases means DNA strands are not strong enough to hold the strands together. DNA ligase enzymes. Okay, these DNA fragments by forming a phosphodiester bond between the phosphate group of 5' carbon of one deoxyribose with the hydroxyl group. Okay, 3' carbon of another deoxyribose. It is very important. Okay, ligase enzymes. Okay, this is the definition. Okay, first scientist name. Okay, first RDNA technology, recombinant DNA technology. Scientist names is very important. Okay, Boyer and Cohen. Next, what is the plasmid? Okay, next, what is the restriction enzyme? What is the ligase enzymes? Okay, what is the plasmid DNA vector? What is the host? Okay, next, clone. What is the clone? It is very important in our DNA technology. Okay, next definition of RDNA technology. Okay, how do you understand? Look at here. The joining together of DNA molecules from different organisms and inserting it into a host organism to produce new genetic combination that are of value to science, medicine, agriculture, and industry. Okay, this is definition of RDNA technology. Okay, how do you understand? Any doubts? Okay.